Today we'll understand the compartmental pharmacokinetic models using simple examples. So the pharmacokinetic model gives us an easy way to express mathematically how a drug moves inside the body over time. They help us to calculate important pharmacokinetic models like volume of distribution, clearance and more. So, question arises, why do we need these pharmacokinetic models? Let's see. These pharmacokinetic models helps us to understand the drug behavior in patients, to calculate the correct dose, to predict drug levels in blood or in other fluids, to check for toxicity due to drug accumulation, to link plasma levels with response, to compare brands or formulations called as bioequivalence, to predict multiple dose results from single dose, to study how diseases affect drug distribution and disposition, and lastly to explain drug interactions. We required these pharmacokinetic models. So there are three main types of pharmacokinetic models. First one is compartment model, second one is non-compartment model, then lastly we have physiological model. Before going to the compartmental model, let's see what do you mean by compartment. A compartment is a theoretical space in the body where the drug is assumed to be evenly distributed. Okay, so a compartment is a theoretical space in the body where the drug is assumed to be evenly distributed. It's not a real organ, just a mathematical area used to describe drug movement and to predict, represent the processes such as absorption, distribution, metabolism, and excretion. So the compartment modeling is a commonly method used to estimate the pharmacokinetic characters of a drug such as ADME. These compartments are hypothetical in nature, and we divided the body into one or more compartment to track how the drug enters, distributes and leaves each one. These compartments can be in series or parallel and communicate with each other. Each compartment act as like a tissue or group of tissue. Okay, tissue or group of tissue with similar blood flow and drug affinity. And every organ or tissue or body fluid that can get equilibrate with the drug is considered as a separate compartment. Drug movement between compartments follows first order kinetics and measured by rate constants such as K12 for central to peripheral compartment and K21 for peripheral to central compartment, which is reverse. There are two main designs for compartmental model. First one is mammillary model, then we have catenary model. Mammillary is most commonly used, catenary is less used. Let's focus on mammillary model. It consists of one or more peripheral compartment connected to the central compartment. Okay, so central compartment, it is denoted by one. We will see in the diagram. Central compartment is comprised with plasma and highly perfused tissues like kidneys, brain, lungs, liver and heart. The drug is directly absorbed into the central compartment, that is blood. Elimination also occurs from this compartment only by liver, kidneys. Now let's see peripheral compartment. Peripheral compartment consists of tissues like low blood flow, okay, like adipose tissues, muscles, skin, bones. Let's see these models. First one we have model number 1, which is one compartment open model with IV infusion. So if infusion means there is no absorption, only elimination will be there. So it is our central compartment. From central compartment the drug is eliminating as it is an open model. It is an open model. Then we have model 2, one compartment open model, extravascular administration like oral, rectal, etc. So extravascular means there will be absorption. So our drug is entering by absorption into the central compartment, then eliminating with Ke absorption, a elimination rate constant. Then we have model number 3, two compartment open model with IV infusion. Two compartment and IV infusion. IV infusion means no absorption. Okay, so no absorption. Only two compartment open model. Open model means elimination will be there. Two compartment means central compartment, peripheral compartment. From central compartment, drug goes into this peripheral compartment. From peripheral compartment again comes into the central compartment. Then elimination takes place from central compartment. Then we have model number 4. Two compartment open model extravascular administration. Extravascular means absorption will be there. So Ka, then our drug enters into the central compartment, that is 1, and from 1 it goes to the second compartment, that is peripheral compartment. So K1 2, then we have the drug will move from the second compartment, peripheral compartment, to central compartment. So K21, then from central compartment it gets eliminated. Model number 5, we have 3 compartment open model IV infusion. Now here we have the 3 compartments. Our central compartment is here 1. So it is, as it is a IV infusion, no absorption, okay, no absorption will be there. So no Ka. So from one compartment, first compartment, sorry, central compartment, drug goes into the second compartment, which is peripheral compartment, then comes back. And as it is a central compartment, 3 compartment model, the from central compartment the drug goes to the third compartment also. So K13, and from third compartment it comes back. So K31, then elimination takes place because it is an open model. Then we have model number 6, 3 compartment open model extravascular administration. So here we have to include absorption rate constant to the central compartment, and as it is, it will be the same with the IV infusion. Okay, the drug goes after absorption, the drug goes to the second compartment, then comes back to the central compartment and goes to the third compartment, then comes back reverse to the first compartment, central compartment, then gets eliminated. So the box 1 is known as central compartment and box 2 is known as peripheral compartment. In one compartment open model, the drug is both added as well as eliminated from the central compartment only. In two compartment open model, the drug moves between the central and peripheral compartment. The number of rate constant appearing in a particular compartment model is given by R. To know how many rate constants R a model has, for IV model R equals to 2n minus 1. For extravascular administration, R equals to 2n, where n is the number of compartments. Now let's quickly understand the catenary model. This model is designed like a train, compartments in a line. Each compartment connected one after the other in series. Just like a train moves from coach to coach, here the drug moves step by step from one compartment to the next compartment, then to the other compartment. But this model is rarely used in real practice. Why? Because in our body, most organs are connected directly to the blood or plasma. They don't follow a strict chain pattern like this. So even though it's look neat, the catenary model does not match human anatomy, which is why we all mostly always prefer the mammary model. Let's see the merits of compartment modeling. Compartment models visually represent the various rate process involved in drug disposition. They also show the number of rate constants we need to describe the rate process clearly. Then third, this helps us to design proper dosage regimen for patients. Then fourth, we have that it has helped us to predict the drug concentration in both normal and disease conditions, pathological conditions. And mathematically, we can track the drug concentration changes in each compartment over the time. Over the time period, we can calculate. Let's see the limitations of compartment models. These models are hypothetical, meaning they don't exactly match real body structure or physiology. Also, the same model not work for all the patient or study groups. And every model is drug specific. It cannot be applied to all drugs. Creating a perfect model needs a lot of effort to match the ADME process. Plus, the equations used are mathematical complex. Mathematically, they are complex. They are multi-exponential equations. A drug might behave differently based on the route of administration, requiring different models. And finally, there are challenges in interpreting results, especially when comparing human and animal studies. And that's all about compartment models in biopharmaceutics and pharmacokinetics. In the next video, we'll explore non-compartment models and how they differ. Make sure to like, share, and subscribe if you found this helpful.